You've probably heard of GMOs, organisms that have been genetically modified to be bigger, more nutritious, or resistant to droughts and pests. Or perhaps you or someone you know has diabetes and takes insulin that was mass produced by yeast in a giant vat called a bioreactor. You may have even heard of Casgevy, the first ever CRISPR-based drug that treats sickle cell anemia by directly editing the genes in patients' blood cells. These are all examples of synthetic biology in action. Synthetic biology is the application of engineering principles to biological systems, designing solutions to real-world problems. To be succinct, synthetic biologists create tools out of the biology of our world.
But what's so great about synthetic biology? I mean, just look at how complicated a cell is, and there's still so much more we don't know about their different processes. Plus, living things tend to die. It just seems so much more unreliable to try to use biological parts instead of human made tools.
Biological systems can be challenging to control, but there's no need to micromanage everything going on in there. Synthetic biology is so powerful precisely because of what we don't have to do. The dynamic nature of biology allows us to outsource work. For example, if our machine of choice is a bacterial cell, if we wanted to upscale, our machine could simply duplicate itself, whereas it would take a lot more time and resources to produce copies of an inorganic machine.
In addition, the materials required to construct biological parts are easily available on Earth. They don't require high temperatures, high pressure machinery, or toxic reactants. Instead, biological parts tend to assemble and operate best at room temperature, atmospheric pressure, and in aqueous environments. Inorganic materials such as metals must be mined, extracted, and processed, using up a lot of energy, placing a huge burden on the environment and jeopardizing the health and human rights of those working in these conditions. Synthetic biology allows us to create parts and systems to address problems that traditional engineering disciplines struggle to solve on their own, expanding the toolbox for engineers to build a healthier, safer, and more equitable world.
But how do synthetic biologists even begin to modify biological parts? Biology has progressed as a series of random evolutionary steps through billions of years of Earth's history. There is a vast trove of genes and biochemical processes and cellular communication pathways, and there's still so much researchers in biology have yet to understand and uncover. To achieve clarity in the face of all this biological information, we as synthetic biologists need to keep in mind three core principles: modularity, abstraction, and standardization.
Different components of a car perform different functions, and thus can be switched out and modified for different user experiences. For example, if we give this car winter tires, its ability to traverse snowy terrain improves. We could also give this car off-road tires if we plan to drive on rough roads. Modularity in synthetic biology works off a similar principle. The different genes, proteins, and cells that you work with are modules that can be independently modified and reused without interfering with other modules. For example, we can insert the green fluorescent protein module into the yeast cell module to create a yeast with green fluorescence. Perhaps the green fluorescent protein isn't to our taste, so we can switch it out with red fluorescent protein, or M-cherry instead. Now, let's say a scientist comes along and wants to use this system to detect when a package of food might be contaminated with foodborne pathogens by turning red when the package gets too warm. They can then add a temperature-dependent promoter or heat shock transcription factors module to the yeast. This module will activate the red fluorescence at high temperatures. As you can see with this example, modules can be swapped in and out depending on your intended function. By doing so, a simple process like a cell becoming fluorescent can become applicable to more situations.
Of course, when speaking about parts like red fluorescent protein, there is a lot of information that we're ignoring. For example, the amino acid sequence of the red fluorescent protein, the 3D structure of the protein, the temperature or pH ranges that this protein is stable at, and the physical properties that allow it to fluoresce. Even though we've ignored all this important information, it wasn't necessary to understand what this machine does and the role that the red fluorescent protein plays. In fact, listing out all 236 amino acids would probably be extremely distracting and make it much harder to organize or modify the system. This is the role of abstraction in synbio. Abstraction allows you to focus on high-level function of biological parts, rather than getting caught in trying to understand the nitty-gritty details. Without abstraction, trying to work with synthetic biology would be as convoluted as trying to remember what you had for breakfast, starting with the average volume in a sip of water. You're saving a lot of time, energy, and comprehension by simplifying to the essentials.
Have you ever heard of the Mars Climate Orbiter incident? The Mars Climate Orbiter was a probe that was completely destroyed because of a simple mistake. The engineers forgot to change their units from imperial to metric. Standardization in synthetic biology makes for reliable and repeatable experiments and devices to avoid a disastrous mistake like the Mars Climate Orbiter. When a synthetic biological part or technique is standardized, that means there is detailed documentation describing procedures, guidelines, and other pieces of information that is universal and should be followed amongst all researchers. For example, let's say our food spoilage detection system was built using the K12 strain of E. coli. A specific bacteria strain is commonly used in research. If another scientist building the same system fails to adhere to the principle of standardization and uses some random strain of E. coli, they run the risk of that E. coli containing a gene that interferes with the heat detection, or even that it could be pathogenic and cause food poisoning. We reduce that risk of failure that comes from variation in biological parts by creating and following standardized procedures. With more predictable experiments also comes the ability of mass production, which is essential for commercial use of synthetic biology products.
Biobricks are DNA sequences with defined functions that combine the principles of modularity, abstraction, and standardization. Biobricks are extremely useful for giving you the framework to build your synthetic biology systems. Assemble the right biobricks, and you can design almost any device. For example, when implementing our food spoilage detection system, we'd start by choosing the biobricks we want to use. Our main function is fluorescence when the environment gets too warm, but we can split this up. We need a system that detects heat, and we need a system that fluoresces. Breaking it down even further, our heat detection system needs to detect when the environment is hot and also trigger the fluorescence. Those will be the functions of our biobricks, one for heat detection, one for triggering fluorescence, and one to fluoresce. We can search the registry of standardized biological parts for the biobricks that we need. For heat detection, we use a DNA sequence that codes for a heat shock transcription factor. For triggering fluorescence, we use a DNA sequence that the heat shock transcription factors will bind to promote gene expression. And finally, for fluorescence, we use the DNA sequence that codes for red fluorescent protein. Now that we've selected our biobricks, we can connect them to produce our desired function. So at high temperatures, the heat shock transcription factor will bind to our promoter region in front of the red fluorescence gene, which will cause the red fluorescent proteins to be produced only when it gets hot.
What we have just done is only the first step in engineering a machine, the design stage of the design, build, test, learn cycle, or DBTL for short. The DBTL cycle is a framework for engineers to continually develop and improve their engineered designs. As we have just done, design consists of the conceptual work. What is the goal of our machine and how will we achieve that goal? What host organism or chassis are we using? What parts are we using? What experiments do we need to conduct to test the effectiveness of our design? What is our predicted results, and can we find previous experiments and datasets to support our reasoning? The next stage is build, where we construct our planned design in the lab. This would include DNA assembly and growing our chassis organisms. Issues in the build step can serve as indications to re-examine and improve upon our design. Now that we have a constructed machine, our next stage is to test how well our design works through experimentation. We ideally want quantitative data from our experimentation so we can analyze the performance of our design in comparison to our expected results, other existing solutions, or previous designs we've tested. There are all sorts of experiments we can use to analyze our design, so you must choose carefully during the design stage to perform relevant experiments. The last stage of the DBTL cycle is learn, where we analyze our design from the test stage and comment on what we have learned from our current design. This may include how to troubleshoot certain issues, how modifying a certain parameter affects experimentation results, etc. This feeds back into design and taking what we've learned to improve and optimize our design. Though a full-scale project would demand many iterations of the DBTL cycle, MiniGEM would be focused on this design stage.
The central story of molecular biology revolves around a few main characters: DNA, RNA, and protein in the central dogma. Genetic information, in the form of a nucleotide sequence, is stored long-term in DNA. DNA is transcribed into messenger RNA, or mRNA, a less stable transporter of genetic information. mRNA is then translated into proteins, the workhorses of biological systems whose functions range from forming cellular structures, to attacking pathogens, to creating energy for the cell. There is a lot more to say about the central dogma, but most synthetic biology projects focus on creating some product using the central dogma.
Synthetic biology is usually categorized in two ways: genetic engineering and protein engineering. Genetic engineering involves the manipulation of DNA to modify a gene or its function. There are a wide variety of tools and techniques used in genetic engineering. Commonly, in bacterial models, genetic engineering involves the construction and insertion of bacterial plasmids. These are circular DNA sequences that the bacteria can transcribe and translate to create proteins. This is how yeast cells are able to produce human insulin for medications. A plasmid carrying the gene that codes for human insulin was inserted into yeast, and the yeast was able to mass produce the protein. Genome editing, which involves changing the chromosomal DNA of organisms, is a subcategory of genetic engineering, which allows for more permanent genetic engineering in cells without relying on plasmids, meaning human cells or agricultural crops can be directly modified. CRISPR-Cas is a system widely used for genome editing. The Cas class of proteins are proteins that can cut DNA and are naturally used in bacteria for eliminating viral genetic material. The CRISPR-Cas system uses a guide RNA which matches precisely with the DNA site you want to cut into. When the guide RNA situates the Cas protein at the right location of the DNA, the Cas protein cuts, allowing for changes to the genome to be made. Depending on the type of Cas protein, you can make changes to either double or single-stranded DNA or make different types of cuts. The other main category of synthetic biology design is protein engineering, which focuses on modifying and optimizing the function of proteins. Protein engineering is more useful than genetic engineering in cases where a protein may not even exist for the function you're looking for. For example, Taxol is a chemotherapy drug produced by the Pacific yew tree. However, the tree only produces small quantities of Taxol, and the yew tree's enzymes involved in Taxol production are not very efficient. However, using protein engineering techniques, we can create new proteins with improved qualities and even ones that perform completely new functions. There are two main branches of protein engineering: rational design, which uses existing knowledge of protein mechanisms and computational tools to create new proteins, and directed evolution, which involves inducing mutations in your gene of interest and measuring which mutations best improve function. Both techniques are time-consuming and challenging to pull off, but they allow us to further expand what is possible with biological parts.
The goal for MiniGEM competition is for you and your team to design your own synthetic biology project by the end of this week. We hope that you will have something like the food spoilage detection system example, a collection and assembly of biological parts to address a real-world issue. The first step to any design project is to choose a topic. By the end of the ideating phase, you should have a one-liner for your project explaining the purpose and which tool or technique you're using. For example, treating pancreatic cancer using crust-based protein. Mosquito sticky trap made with recombinant biolarvicidal toxins and beetroot peel attractant. Or antidepressant cookie with bacteria producing antidepressant compounds. Here are some approaches that we have found helpful when ideating our own projects.
A top-down approach starts with identifying an issue that you care about. We've given you a list of global issues in the synbio toolkit to help with this. Identify root causes of your chosen issue, current methods of addressing this issue, and past iGEM projects that relate to this issue. Be sure to focus on the gaps that exist in current solutions. The next step is to define your project goals. When using the top-down approach, you might find difficulties in properly narrowing the focus of your project. As an example, early detection of cancer is an extremely broad topic that is hard to understand and address, so it's best to add specificity. It's far easier to brainstorm solutions for a specific goal rather than trying to tackle a vague issue. So instead of trying to find a method for early detection of every human cancer, it's much more realistic to find a solution to increase accuracy for early breast cancer detection. Next, identify possible biological parts that might be able to address these gaps. It can be challenging to find a novel application of an existing part to your issue of interest, so this is a good stage to consult the synbio toolkit and ask for opinions from experts or advisors to guide your research. There isn't an easy protocol to follow for novelty, so have confidence to pursue your ideas seriously, and remember that you're not looking for a perfect solution,
but a solution that improves upon certain aspects of existing solutions.
A bottom-up approach starts with a cool part or technique you find interesting. This is equivalent to choosing a cool protein from the synbio toolkit. We recommend that you read recently published papers to understand cutting-edge developments in the field of synthetic biology. The benefit of this strategy is that there's more opportunity for finding a novel application, though it may be more challenging to work with than with a less well-established part or technique. Once you have chosen the part or technique you'd want to apply, be sure to read the background literature on how this part or technique works and unique properties. This is an essential step to exploring possible applications for your part. Finally, once you've gotten a grip on what makes your part unique, look for potential applications for those unique qualities to shine. See if you can optimize existing applications or use your creativity to come up with completely new applications. Some papers may even have a future direction section to give you inspiration for new applications.
Now that you've settled on a topic, it's time to determine exactly how your solution will work. When stepping into research, you will encounter the phrases wet lab and dry lab. These are simply two different, yet synergistic approaches to developing a research project. The wet lab entails hands-on experimentation with cells, chemicals, and physical samples, whereas the dry lab entails conducting data analysis and making predictions through computational and mathematical models and simulations. We'll be asking you to create a plan for both your wet lab and dry lab for your chosen topic.
For the wet lab, there are two major design questions you need to answer. First, you need to determine what biological parts you are using, the materials. This would include a chassis, which is the host species and strain of cells, biobricks, DNA sequences coding for specific functions, as well as relevant proteins or products. The second part to constructing your wet lab plan is to explain how you will assemble your system, the procedure. Now that you have your materials, how do you assemble them? Your main consideration should be how the assembly of your parts serves your intended purpose.
To find parts and techniques that fit your project, it may be helpful to refer to the synbio toolkit for a list of interesting proteins and organisms. You should also check the materials and procedures section of the experimental papers that inspire your project. You'll find details on what parts and procedures the researchers use to conduct their experiment.
The dry lab design is usually more flexible compared to wet lab design, as dry lab projects support and supplement the wet lab experimentation in synthetic biology. For a successful dry lab design proposal, you should first decide what the purpose will be. You may choose to simulate your experiments with models, creating graphs and predictions given specific inputs. You may want to improve the implementation of your design by designing a software or hardware tool that supplements your biological system. You could also choose to improve upon or build a new measurement tool to analyze your solution for different metrics of success. Whatever you choose, your dry lab design should outline what existing resources you're using. This may include software tools such as molecular modeling libraries or protein folding models, hardware tools such as sensors or bioreactors, and other resources such as protein databases.
If you are stuck at any step, it may be helpful to take a step back and reconsider the scope of your project. These are also important questions to consider when discussing your project and why it is important. Who are the stakeholders that may be affected by your project? What outcomes are you hoping for them? What are the current barriers to existing solutions? Why might your solution be better than these alternatives? And what are the barriers that might prevent the success of your proposed solution?
Finally, you've created a preliminary design. However, science is so much more than just achieving technically difficult results. Making others care about your project is crucial for using technology to improve lives. After all, who's going to benefit from your technology if no one's convinced to use it? By the end of the week, if you wish for your team's project to be considered for a prize, submit a project proposal explaining your project design process in any medium you want. This can be a scripted video, an infographic, a written proposal, or a slideshow. What matters is that you present a strong case for the impact of your proposed solution and that you and your team have put a lot of thought into your design. Be sure to highlight how your design changed as you learn new information or consider different perspectives. That way, you can demonstrate how each element of your design is deliberate and meaningful.
It may also be helpful to approach your presentation with this year's special categories in mind: entrepreneurship and sustainability.
The entrepreneurship category asks you to define a path to bringing your solution to the market. A successful entrepreneurship plan may include development milestones and timelines, identification of potential investors, and sources of funding, and comparison to existing or developing technologies on the market. To pursue the Entrepreneurship Prize, you need to present a strong argument for why your solution would be successful if commercialized. Convincing others in the future commercial success of your project hinges on selling people that your solution is desirable and that your team should be trusted to bring it to consumers. Here is a suggested approach to pitching your solution. Start by explaining your problem statement and impress upon your audience why this specific problem is so crucial to address. Next, give an overview of the current market addressing your problem statement, including existing competitors and gaps or flaws in the status quo. Describe your solution and place emphasis on how it addresses the gaps you've identified. Next, lay out a timeline for how your idea will be commercialized. What resources or budget do you need at each stage of your development process? When do you anticipate obtaining any protection to your intellectual property? And how will you test your solution? And by what metrics will you determine if the quality is good enough to bring to market? Consider creating a Gantt chart outlining your business plan like SURF PFAS from 2024 University of Padua Adjunct Team. Good luck!
The sustainability category asks you to define how your solution fulfills the United Nations Sustainable Development Goals, called SDGs, in its purpose and/or implementation. Sustainability entails ensuring present needs are met without compromising the ability to meet the needs of future generations. Though sustainability is typically thought to be a synonym for environmentally friendly, protecting and preserving natural ecosystems against climate change, pollution, and resource depletion that threaten the future of the environment we live in, caring for natural environments is actually only one dimension of sustainable practices. A sustainable future also requires social stability so that individuals and communities can safely and equitably achieve well-being, and economic prosperity so that people can obtain the resources to survive and thrive. The SDGs are 17 milestones set by the United Nations to build a sustainable future for everyone around the world. If you're pursuing the sustainability category, aim to fulfill multiple SDGs and provide explanations for how your solution contributes to achieving these SDGs. You must explain how your solution addresses the 3 pillars of sustainability: society, economy, and environment, as well as how your design process was influenced by considering the SDGs. It's especially helpful to provide details on the expected results of your solution, identifying who will be benefiting and in what ways are they benefiting. You should also discuss how your solution exists in the context of its application. What potential barriers exist to your solution, and how should you, as a scientist proposing the solution, be working to address these barriers? You can start with an explanation of your problem statement and why this issue is important. At this point, you should also explain the SDGs your solution addresses and its relevance. For example, building a system to reduce fertilizer runoff helps fulfill SDG 14, conservation of life below water, but it's probably a little bit of a stretch to claim it fulfills SDG 4, inclusive and equitable quality education. Next, explain the science behind your solution, what is the impact, and to what extent does it progress your SDGs.
We hope you're just as excited for MiniGEM as we are. We know this is a lot of information to kind of just throw at you in 15 minutes, and we also understand that it can't always be easy to learn everything from reading articles or using online resources. We want to let you know that the best learning opportunities do come from good conversations. So we strongly encourage you to talk to our mentors and bounce your ideas off of them. We encourage you to look out and seek out external sources, and even to just talk to your peers and see what ideas they might have. We're really, really excited to see what you are going to create, and we wish you the best of luck with this.